Indian dwarf wheat, which is also known as Triticum sphaerococcum, for future wheat improvement programs. So most of you might be PhD students or say MSc students. Some of them are in some of their faculties as well. Regarding the Triticum sphaerococcum, this is somewhat distinct from the bread wheat, which is Triticum astribum, and it looked like this. If we see our normal wheat, the spikes are somewhat longer, about 10 to 15 centimeter long, but in Triticum sphaerococcum, they are very compact, and the seeds are round in shape. Okay, so if you have seen the seeds of wheat, it is oblong shape, slightly larger, plumpy. But in case of Triticum sphaerococcum, the seeds are very spherical one. That's why the name comes as Triticum sphaerococcum. Okay. So about the introduction, this wheat, Triticum sphaerococcum, it was belonged to India. Okay, so it has Indian origin, and this was a staple crop in India and Pakistan from the Bronze Age. Means from last 3,000 years, it was under cultivation in India and the Sindh region of the Pakistan. Okay, the archaeological evidence suggests that in the Mohenjo-daro excavation site, seeds of this species have been found. Okay, and based on the carbon dating, they have came to know that yes, this species was under cultivation from the Bronze Age. Okay. Then what happened? Because nowadays we see very less or almost extinct cultivation of this species, and this has happened because the onset of the Green Revolution in 1964. So from last 3,000 years, it was under cultivation, but as we got high yielding varieties from Summit, Mexico, the Green Revolution happened in 1964. The cultivation of this species was stopped, and it's obvious because those high yielding those varieties which were dwarf, they were higher yielding than this sphaerococcum type and sphaerococcum type, and these species was not cultivated thereafter. Okay. So being a landrace, this sphaerococcum wheat, it is now almost extinct from the cultivation. Therefore, it has not been studied in detail for morphophysiological trait as well as the molecular level, maybe SSR or SFP markers. The studies are very scarce. Okay. Now, our Indian National Gene Bank, which is in NPPGR, they are holding some of the accessions of the species, but I have collected the accessions from the major gene banks, and they are very, very limited. It means the genetic diversity present in this species is almost lost. And if we lost this species, then we will lose heritage species of this India. Therefore, its conservation, characterization, documentation, and its utilization in the breeding program, it is very much needed. Okay. So these ancient wheat species, which was neglected for a long time, it has the potential to contribute useful alleles. And these useful alleles, they are in the terms of tolerance to abiotic stress and as well as the nutrition need. Okay. So this is just an example where in the top corner we could see the Triticum astribum, a plant type, the spike as well as the grains, okay, which normally we see in the field. But it has suddenly changed to this plant type. Okay, the spikes become compact, the height become reduced, and the grain shape was totally changed. So this has happened only because of two missense mutations, and that mutations were located on the 3D chromosome of the wheat. Okay, and that locus where the two missense mutation happened, it was termed as S1 locus. S1 stands for sphaerococcum. So these two mutations, so you could see the power of mutation, what mutation could do. Mutation could create new genetic variation. And this is the perfect example where we could see two missense mutation has leads to the development of extremely new phenotype. Okay. Now, why this dwarf wheat with small grain and low yield potential, it was still under cultivation in India and Pakistan. Okay. So before that, I would like to share that this S1 locus, it encodes a protein, which is the GSK3-like kinase, and these two independent mutation in the T domain that leads to the complete change of the phenotype. Okay. Now we know that this this phenotype, or say this species, it was under cultivation. Our ancestors, they have conserved this species for a specific purpose. Okay. Some archaeological hypotheses that these small grain types that were selected, which suit the culinary needs at that time. Okay. But the main thing which has happened, that these the earlier farmers, or say earlier our ancestors, they have conserved this species because it has a tolerance to drought stress, it has a tolerance to the heat stress. Okay. Also, the grain quality of this species, it is very good. It has a very high grain protein content. It has high iron and high zinc as compared to the our cultivation, our cultivated wheat. That's why this species was under selection from at least 3,000 years till now. Okay. Now, what we did at IRA, we have collected the accession from the major gene banks of the world, from USDA, Germany, then Bulgaria, and some of the accessions were contributed by the Agriculture University at Punjab, PAU, and some of the accessions they were collected from the farmers. So, in total, around 116 accessions we got at a time. And this is the largest set of accession known at present anywhere in the world, and which is present in the ILM. So our target was to evaluate these accessions for the drought tolerance, heat tolerance, and we would like to see whether it is really showing tolerance to terminal heat, tolerance to drought, or does it have really the high grain and high nutritional value. So we have evaluated these accessions under 3 environments for more than 2 years. It was time zone irrigated, which is optimum condition. We have grown it in the restricted irrigation condition, which mimics the drought tolerance, because wherever there is a drought, wherever there is irrigation, 1 or 2 irrigation, generally wheat is grown. Okay, not complete under drought condition. Therefore, the RIM, which is restricted irrigated conditions, we have followed. And of course, we have followed the late zone condition for the terminal heat tolerance. So this is the box plot, which shows that actually we have evaluated for the morphological traits, physiological traits, and the nutritional quality traits. I have not shown everything, but I have chosen some important traits to show. So this red one, they are the late sowing. The green one, they are the timely sowing. And the blue one, which is the RI condition. Now there are total 6 box plot. First represent the sphaerococcum. First represent the astivum, and the second one represent the sphaerococcum. Why we have compared? Because the sphaerococcum, it was originated from the astivum, but because these are the two different species, the comparison is not that much valid, but it shares the common genome, right? BBAADD. Both astivum and sphaerococcum have the same genome. That's why we have taken this species for the comparison. And we could able to see for days to flowering, obviously because this is a land race, it has a longer duration as compared to the astivum one. For the NDVI, which shows the greenness index for canopy temperature, at 3 different stages we have taken, and we could able to find that the canopy temperature of these species, particularly under the late zone condition and RI condition, it is lower as compared to the astivum. It means it could able to cool your canopy at the stress condition, and that has leads to the somewhat higher grain yield under terminal heat as well as under the droughts condition. When we have compared the grain, iron, zinc, and protein also, it is significantly high, reaching up to 16 to 17%, and grain iron has reached up to 50 ppm, and zinc has reached up to 60 to 65 ppm. Where in the traditional bread wheat, we could able to see the protein content from almost 9 to 13%, maximum 13%. Grain iron and zinc, it is around 40 to 45 ppm. That's it. But this species has very high grain content, very high iron and zinc content. Now we have compared the yield. This mm, this color, brown color entries, they are the checks or separate bit varieties, and these are the some of the varieties which we have taken for the yield evaluation. And we could able to find some of the lines which are comparable in yield as compared to the estimum, but none of the line which is higher yielding than the Triticum estimum or separate bit. And this is obvious because this is a land race, and we cannot compare a land race with the variety which is developed by breeding. Okay, because it always have a higher grain yield. So, but in that also under stress condition, under RI and under late zone condition, we could able to find some of the genotypes which are comparable to that of the estimum types. And we have published this study because this was the first report, and no one has done it. It was published in the Genetic Resources and Crop Evolution 2024. Okay. Now we know that this species having tolerance to terminal heat as well as the tolerance to the moisture stress condition. I will not say drought stress, I will say it is a moisture stress condition. So the same environment data, yield data we have used, and we have estimated the 10 heat and moisture stress susceptibility indexes. These index, they are very common, and they are not new. Okay. But when we have found that the heat susceptibility index of almost 50% of the entries comes under the tolerant category. Okay, so means that most of the lines in that panel, they were having tolerance to the terminal heat as well as the tolerance to the drought stress condition. Even 43 lines, they come under the highly tolerant category based on the DSI or say HSI. Now, these are these indexes which we have followed for our paper, and they're pretty old. This is not a new thing, but because first time we have estimated these indexes in these species, our paper got published in the scientific reports. So these stress susceptibility index, the stress susceptibility percentage index, and percent yield reduction. These are the three main important criteria which by which we can observe that okay, our species is tolerant to heat or tolerant to drought or any stress condition. So we required a low value of SSI, low value of SSPI, and low value of PYI. Also, the relative stress index RSI, these values should be low, but we need a high value for yield index, high value for yield stability index, and some of the parameters which are related to the productivity. So positive values for some of the traits and negative values for the traits related to SSI, SSPI, RSI, and PYI. These could easily tell you that okay, these are the lines which are resistant to tolerant to the particular biotic stress. So this is the ridge plot which shows the yield of the Triticum's sphaerococcum as well as the Triticum estivum at normal condition, at heat stress condition, and at the RI condition. And these are the 10 heat and drought susceptibility indexes. So the first one is SSI. If you could see the top ones, they are the Triticum sphaerococcum, and the bottom they are the Triticum estivum. Okay, braid wheat. S stand for heat, D stand for drought. Okay. If we could see the mean values, it is below 1. So the heat or drought sensitivity index is range from 0 to maybe it is more than 1. So 0 to 5 it comes under tolerant. 0.5 to 1 it is come under moderate tolerant, and beyond 1 it is susceptible. So almost most of the entries they show the tolerant reaction for SSI. Similarly for SSI, SSPI, which clearly differentiates sphaerococcum from the estivum with regards to their value. If we could see the bottom graph, which is percent yield reduction, in the estimum type, we could able to observe the yield reduction from almost 15% to 45%. But in case of sphaerococcum, we have observed from 5% to almost 20%. So this species, no doubt, has the ability to tolerate moisture stress ability to tolerate heat stress. And we could able to identify some of the genotypes which are really showing the good performance under timely heat, timely late, and the drought conditions. Okay, then we have published this paper in the scientific report in 2025. Then our next target was to assess the genetic diversity and population structure, and this was the first report. No one has done it. So these Triticum sphaerococcum accessions were genotyped with the 35K SNP array, and from the 35K we could able to take out around 11,680 highly informative markers, and these markers were later used for the assessment of genetic variability. So we have estimated the nucleotide diversities, the GMAS D statistics, PIC content. The population structure was also worked out with the help of TASL, and those markers which are having peak value more than 0.55, 0.15, they were taken for the analysis of AMOVA, the genetic diversity and principal coordinate analysis. We have also done the linkage equilibrium, LD mapping, and haplotype block analysis was also done with the help of the around 11,000 informative markers. So regarding the genetic diversity analysis, as we know, it has three genomes, A, B, and D. And the high GD value, if you could able to see 0.21, 0.22, and the high PIC value, 0.18, 0.19, around that. Of this panel, it indicates a greater genetic variation. We have also worked out the GMAS D statistics. Basically, this statistic, it is highly popular in the population genetics analysis. So this is basically the difference between the two measures of genetic diversity. The first measure is mean number of pairwise differences and the number of segregating sites. So these difference leads to calculation of the GMAS D statistics. If the GMAS D value is 0, it means the observed variation, it is similar to the expected, and there is no evidence of the selection. If the GMAS value D is less than 0, it means there is abundance of rare alleles or say excess of rare alleles, and it shows that some selective the population structure has been happened. And if the GMAS values, they are more than 0, and in this case it is 0.4, which is very high, it means the balancing selection has been done. It has been done by the previous farmers or our ancestors who have grown this wheat from last 3,000 years, and it shows that the rare alleles, they are very limited, but it has abundance of positive alleles for the trait of interest, and that time the trait of interest were the tolerance to abiotic stresses and as well as the better nutritional content. So around these, the filter SNPs, they were also used for the phylogenetic analysis, and this green analysis, the phylogeny tree-like structure, also of Triticum aestivum, and red belongs to this Triticum sphaerococcum. So the same 35K panel clearly differentiated Triticum sphaerococcum from the Triticum aestivum. Then we have worked out the population structure, and the delta K value was 4. It means in our panel there were 4 subpopulations. Okay, and these four populations were also confirmed by the principal coordinate analysis. Then we have worked out the allelic pattern indices, the genetic differentiation studies. We have worked out the FST values. So FST value basically provides the better estimate of genetic differentiation. So high the FST value means high the fixation index. So this subpopulation 1, 2, and 3, they have the FST value from 0.6 to 0.7. It means that most of the alleles in this population, they are fixed. But in case of subpopulation 4, it is 0.4, which is very less. It shows that there are still variation observed in the SP4. Okay. Then we have worked out the LD linkage disequilibrium, and the LD size, LDDK block was around 7.59, because the population was very small, 416. The size is a little bit bigger. Then we have worked out the haplotype block analysis also, and we could able to find that the subgenome B, it harbors highest number of haplo blocks as compared to subgenome A and subgenome D. It is because subgenome B in wheat, it is the oldest and the maternal. Okay, so the D genome came very late in the evolution. Therefore, the D genome, they are having very high haplotype block size, whereas in the subgenome B, it is very small. And it is only because the more recombination event has happened in the B genome as compared to D. Therefore, the block size, it was small in case of B as compared to A and D. Okay. So this was the first report, and we could able to publish this finding in the BNC Plant Biology. And a good number of citations they are coming from this study. Then we have also worked out the nutritional parameters of these species. The same panel, it was subjected to the protein, iron, zinc, as well as the gluten properties. Okay. So Jiva study was done with gluten parameters. It was estimated by glucometric methods. Gelda method was used for protein, and EDXRF method, which is quite standardized for the iron and zinc. So we have used the multi-local GOS model, and from the 2 years of testing, we could able to identify some of the MTAs which are which shows at least presence of two models over the 2 years. Okay. And then we have used the candidate gene analysis also just to confirm our results. So these are the some phenotyping of the grain iron and zinc content. If you could see the sphaerococcum lines, the average zinc content, it goes up to 53 ppm, and grain iron content is average, I am telling, it is 41%. But in case of the estivum time, estivum, it is 35 and 38. So for grain iron content, there is 8.6% increase in sphaerococcum as compared to estivum. But in case of grain zinc, it is 48% high. So you could see why our ancestors, they have preferred this species, because it has a very high high zinc content over the traditional estivum type. So the marker trade associations were worked out, and we could able to find some of the clear triplicutians which shows the association with more than one trait. Okay, and when we have done the putative candidate gene analysis, we could able to identify some of the genes which are actually related with the protein content, related with the grain iron and grain zinc content. So further validation of these markers through CASP, it is under progress, and very soon we will come up with the publication on this. So these are the silent finding of this study. There is 8.6% high iron, 14% more zinc. Then we could able to identify 55 reliable QTNs. When in silico analysis was done, we could identify some of the QTNs. They are also associated with the genes which codes for the protein content, high iron content, and the zinc content. So these manuscript we have submitted in last week, and most probably it will go through. So we have also worked out the some of the amylose content of this in this species, amylose pectin, and we have worked out the glycemic index also. So this is the this also the first study. And, I will not disclose some of the data because we have recently submitted the paper. Once it will get published, the data will be open to all. So we have also seen the market rate association, and we could able to identify many loci, many QTNs, which are consistent over 3 models for the 2 years. So very soon we will publish these results as well, and we have submitted this paper to the journal Food Chemistry, which has very high impact factor of more than 9.4. And hopefully it will get published because this is the first study. So what are the future strategies? These improvement in the sphaerococcum for the rust resistance need to be done, because these sphaerococcum lines cultivated since long, now they are susceptible to the rust diseases. And for the any variety to go into the farmer field, it should have the better rust resistance. So we are planning in this way to incorporate the rust resistance of the gene. Then we have identified the donors for the nutritional quality traits, and we are in the process for the mapping of these loci through RILS as well as through QTLSET. So our next target would be the mapping of these traits for the quality traits. Also, we are introducing the novel variability for basically for the nutritional traits, because we could able to find the traits related to biotic stresses, sorry, abiotic stresses like heat and drought. We observed it in the bread wheat as well as bread wheat also. But for the nutritional quality, the range is very low. And now the in the area of biofortification, we need to enhance the nutritional content of the bread wheat. Therefore, we could able to introgress the some of the traits from sphaerococcum into the S2, and also these lines could be registered at the genetic stocks. So this was all about small efforts which we are doing at IRI for the integration of novel variability from sphaerococcum to the Astrium. Okay, thank you so much, because on very on short notice, I'm present.